All right, so chapter 9, cellular respiration and fermentation. So in metabolism, which we introduced in chapter 8, we kind of go through the basics. Chapter 9 and chapter 10 are taking two metabolic pathways, cellular respiration, chapter 10 is going to be photosynthesis, and taking these metabolic pathways and really digging into them and trying to understand how living organisms harness energy. To fight entropy, right? So cellular respiration is going to be super important both in animals and plants, and that is something I want to point out. We'll mostly talk about animals in this chapter, but realize plants also have mitochondria. Plants store the energy they make through photosynthesis in the form of sugars, and then they can also use that to run cellular respiration. But we'll primarily talk about cute animals like puffins in this chapter, right? So we're going to talk a lot about that energy transfer. So here we see basically this puffin taking fish, a form of potential energy, right, and turning that into the kinetic energy that he or she needs to fly. So this figure 9.2 is really, really key. So in ecosystems, right, organisms are living in an open system. And so this means that there are inputs from outside. And so the way this works is the input from outside of Earth is going to be light energy, right? Electromagnetic radiation from the sun. And this energy is then going to be harnessed by plants and bacteria to do photosynthesis. Through photosynthesis, they are going to make organic molecules, things like sugars, amino acids, lipids, as well as oxygen. This is a good time to get this down. Oxygen is a byproduct of photosynthesis. And then either those plants or organisms that maybe consume those plants or consumed other organisms that consume those plants can take that energy and put it into the form of ATP. Now, this is interesting. So this light energy is moving into basically this form of an organic molecule, right? So we're storing the energy in the bonds of, say, glucose. The mitochondria is then going to liberate that energy, but most of it wants to repackage into another form of potential energy, ATP, that could then be used as kinetic energy, right, to do cellular work. The key thing, right? Sorry, I got really excited about this image, is that light energy is coming into our ecosystem and energy is going to leave the system in the form of heat, right? So heat kind of exemplifies some of that entropy that we talked about. It's this less useful form of energy. We can't really harness it to do work, and it's just going to slowly dissipate out into the universe, right? Matter is handled very differently in an ecosystem. It cycles, right? So remember, the plant or algae or bacteria is taking in CO2 and water and converting that into another molecule, say glucose and oxygen. And then the mitochondria takes that glucose and oxygen and makes CO2 and water, and round it goes, right? So energy flows in one direction through a system. Matter is going to be recycled in that system. Respiration, a couple things here. It is a catabolic pathway. Again, and the end goal is to produce ATP. Now, cellular respiration has aerobic and anaerobic, how do I say this? When you get to microbiology, you will look at anaerobic respiration more. Typically in here, when we're talking about cellular respiration, we're referring to aerobic respiration. We'll talk about fermentation as well, which can be anaerobic, but typically we just call it fermentation. Okay, we are going to follow glucose through cellular respiration, right? So they're showing C6H12O6, which is glucose, combining with 6 oxygen atoms to become 6 CO2, 6 H2O, and energy, right? And that energy, again, a lot of it is in that form of ATP that the cell wants, but we'll also lose a lot of that energy as heat. So we're going to follow glucose, but I did want to make sure that this was clear to everyone. and we'll talk about it maybe briefly at the end, that you can use fats and proteins as fuel sources in cellular respiration as well. Okay, so a couple things to keep in mind before we really jump in. A lot of what you're going to see in cellular respiration are what are called redox reactions. Redox is a contraction of two words, reduction and oxidation. They made this contraction because the two are always together, okay? So to become oxidized, right, or an oxidation reaction means that a molecule or an atom is going to lose an electron. So oxidation is the loss of an electron. Reduction is the gaining of an electron, right? So you can't gain an electron unless someone else has lost it. And so these two are always paired, right? Oxidation reactions and reduction reactions will happen together. If you need help remembering this, because this does tend to trip people up, right? Oxidation is loss, oil, rig, reduction is gain, right? So oil rig, oxidation is loss, reduction is gain. Now, this is kind of how chemists think about redox reactions, right? Where did the electrons go? What you're going to see, let's see, here's a kind of generalized version, right? So we're showing that electron being lost, right? So X plus an electron is just becoming X. Y gains that electron, and so it's been reduced. Now, cellular respiration, what the biologists are doing is not just following electrons. We are following electrons, but the electrons that we're interested in are on the hydrogen atom. Okay, so don't let that throw you. When they're like, where did the electron go? Look for the hydrogen. So here we see glucose, C6H, follow the hydrogen, H12O6. We're going to lose those hydrogens, right? And so in the process of cellular respiration, glucose gets oxidized. It loses all its hydrogens and becomes carbon dioxide, while oxygen is actually going to go ahead and gain those electrons. It gets reduced to the form of water, okay? And so we're going to follow this equation through. Okay, so to help with these redox reactions, we are going to use some electron shuttles, some molecules that are happy to pick up electrons and then let them go later. You don't have to memorize this whole structure, right? But this molecule NAD+, nicotinamide adenine dinucleotide, right? It's actually based off of niacin, which you might recognize as a vitamin in your diet. And this niacin, right, this NAD+, is willing to become reduced, right, and become a slightly different molecule. I don't like this picture. I think they're just trying to zoom in here, right? And they're ignoring the rest of this molecule. And what they're trying to show you is that instead of one hydrogen, there's another hydrogen. Yay, it gained a hydrogen. So it went from NAD, there's the hydrogen, to NADH, right? So that's the reduction. And what we'll see is later, it's willing to give up those hydrogens. And so NADH is willing to be oxidized. See how this arrow goes this way? And go back to being NAD+. We'll also see a closely related molecule, FAD and FADH. These are based off of riboflavin, another important vitamin in your diet. So just think of these as shuttles. They're going to pick up electrons, carry them around for a while, and then let them go at the electron transport chain. Okay, so speaking of the electron transport chain, the idea here is what we want to do, right, is, how do I say this? So when I think about cellular respiration, right, we're going to look at glucose and oxygen, but this could just as easily be like a chunk of firewood and oxygen, right? And that firewood can burn in the presence of oxygen, right? And it releases all this heat and this light, right? And so you see that release of energy. What we're going to do in living systems is we are going to try to control the release of that energy because we don't want to spontaneously combust. We just want to make little packets of energy called ATP to save for a rainy day. And so what we're going to see towards the end of cellular respiration is an electron transport chain that just slowly kind of passes electrons down a series of proteins to help release the energy in a controlled manner. This will make more sense maybe when we get there, but think about that. This whole process, all these steps, they are set up by enzymes. Okay, so your textbook does a wonderful job of laying out this process of cellular respiration. In fact, I teach cellular respiration in A&P2. I teach it in microbiology. Like, it just keeps coming up, right? So this is definitely one of those things that you should just go ahead and get down. And I will say your book does an awesome job. In fact, I find myself often in those classes coming back and using some of the material from your textbook. So here we go. We are going to look, and I will say this, they're making this into 3 steps. They've color-coded this for you, which is very nice. We will talk about glycolysis first, the splitting of sugar. This second step, I often call pyruvate oxidation, step 2, and citric acid cycle, step 3. If you want it to be 2A and 2B, that's fine. If you want to make them separate numbers, fine. These aren't technically, like, there's no right number of steps you have to have. Same thing. So I usually call this 1, this 2, this 3. So oxidative phosphorylation is 4. And I usually talk about electron transport chain as like 4A and chemiosmosis as 4B. But there's no reason it can't be 4 and 5. Again, so just you're trying to learn this process. It probably makes a lot of sense just to kind of stick with what the book has going, and I will try to be good about doing that just because they did do a color code. But if it makes more sense to you to split it into 5 steps, by all means. Okay, so what we're looking at here, right, and we'll go through this kind of multiple times and we'll get a little deeper each time, so stick with me here. So the first step in cellular respiration, they are pointing out, takes place in the cytosol, right? So outside of the mitochondria, in the cytoplasm, in that fluid, right, the cytosol. Glycolysis takes glucose, which is a 6-carbon sugar, and splits it in half. And literally this word, lysis, means to like split. And glyco should make you think glucose, right? So we're going to split this in half. So pyruvate then is a 3-carbon molecule. So you go from 1 6-carbon molecule to 2 3-carbon molecules, right? We also, in this step, do produce 2 ATP, right? So we already got a little bit of energy. And we also get a couple electrons, 2 electrons being shuttled by NADH. And we're going to take those into the mitochondrion, into the electron transport chain. Okay, so there's glycolysis, right? It's in blue. Very nice. Okay, our next step is we're going to take that pyruvate and we're going to oxidize it. What does that mean? We're going to take a couple electrons off of pyruvate, good. And in fact, we are going to convert it into a molecule called acetyl-CoA. Now, I know this sounds like a foreign language, it kind of is, but acetyl-CoA, what you need to know is it's a 2-carbon molecule. So we went from a 3-carbon molecule to a 2-carbon molecule, which means at pyruvate oxidation, we are also going to begin releasing CO2, right? Because matter can't be created or destroyed. So you can't just go from having 2 3-carbon molecules to 2 2-carbon molecules. The carbon had to go somewhere. It's in the form of CO2. You also, if you notice, since this says oxidation, you do actually reduce more NAD to NADH. And I don't know why they didn't put that in this picture. Maybe it'll pop up later. Okay, and then we hit the citric acid cycle. So the citric acid cycle is pretty complicated, but don't worry, we're not going to go too far in here. But you should realize, right, that we start shuttling more electrons in the form of NADH and FADH. There's the riboflavin-based one. We are going to finish the oxidation, right, complete the oxidation of that glucose. And so during the citric acid cycle, if 4 carbons came in, right, 2 2-carbon molecules, we're going to breathe out 4 more CO2, and we also get 2 more ATP for glucose. Okay. in our third and final step, oxidative phosphorylation. This name sounds really complicated, but it tells you exactly what's happening, right? Oxidative, so we're going to oxidize some stuff. And then with the energy from that oxidation, we are going to phosphorylate things. Remember, a phosphorylation reaction is the addition of a phosphate group. So we're going to take adenosine diphosphate and just start adding phosphates. We're going to make a bunch of ATP. Okay. So, and of course, we'll get into more of the details of how that happens. So maybe the only thing I don't like about this picture is there are, in fact, two NADH coming in to the electron transport chain off pyruvate oxidation. And I like to know where the carbon is, but whatever, we'll get there. Okay, I'll go ahead and post this video on Canvas so you can watch this BioFlix on cellular respiration. Let's go ahead and just jump in and see what this looks like. Oh, I was going to point this out too. So most of our ATP is made in that oxidative phosphorylation, because again, phosphorylation is taking ADP and adding phosphate to get ATP, right? You had 2 diphosphate and then you add a third triphosphate. So the ATP that's made in glycolysis and the citric acid cycle is what's called by substrate level phosphorylation. And that really just means that there's an enzyme that helps bring those 2 molecules close enough together to store that energy in the ATP bond. Okay, throughout your book, again, it's color-coded and they keep adding these little mini maps so that you have an idea of where we are. So kind of keep going back to that, referencing where you are to make sure things are making sense. So we're going to dig in here a little more to glycolysis. Now, to me, the big picture in glycolysis, right, what you really need to know, right, is what's down here at the bottom, right, what the net result is, the final result. So we take glucose and we split it in half, right, into 2 pyruvate molecules. And for me, I want you to know kind of where these carbons are going to. So glucose is a 6-carbon molecule. Pyruvate is a 3-carbon molecule, but of course you have 2 of them, right? So we still have all of our carbons accounted for. Notice we also get 2 ATP and 2 NADH, and they're doing a really nice job of laying out here how that NAD is actually gaining electrons, right? They're with the hydrogen. And so that's the reduction reaction. Now, this ATP, what they're showing you, and we won't spend too much time here, but glycolysis is actually a pretty complicated process. If you start going through some of the images in your book, you'll notice this. But what you do is when you get glucose, to actually like kickstart this whole reaction, you are going to use some of the energy from ATP. In fact, what you need is to add phosphates onto your glucose to help it then run through the reactions, right? So you put 2 ATP in and you get 4 ATP out, but really net is what counts, and there's your 2 ATP, right? So anyway, that's what you want to know. From glycolysis, you get 2 3-carbon pyruvates, 2 ATP, and 2 NADH, right? We're shuttling those electrons. Okay, pyruvate oxidation. Again, we are going to oxidize pyruvate. So here we are, right, zoomed in a little bit more, looking at what happens. So here's pyruvate. Notice 1, 2, 3 carbons. Oh, but look at this. CO2. Look, see, this is what your book is so good. Look at the color coding here, right? So it's already indicating which of the carbons and oxygens we're going to lose in the form of CO2. The remaining molecule, right, is a molecule called acetyl, and it is bound to coenzyme A, and so we call it acetyl-CoA. So in pyruvate oxidation, we take 2 pyruvates, right, because we're always thinking about a whole glucose molecule, and we make 2 acetyl-CoAs. And we also reduce 2 more NAD to become NADH, right? We're going to carry those electrons to the electron transport chain. See, isn't that weird? They didn't show that in the last picture. I don't know. Okay. Oh, and since we've got 2 pyruvates going to 2 acetyl-CoAs, we get 2 CO2, right? So the net from pyruvate oxidation, 2 acetyl-CoA, 2 CO2, 2 NADH. No ATP. It's all right, because we're getting there. So on to the citric acid cycle. So in the citric acid cycle, so it is a cycle. What we're going to see happen here, right, is we take our acetyl-CoA, and they're trying to show you pyruvate oxidation again for some reason, but there's our acetyl-CoA that we just made. Now, when it comes into the citric acid cycle, it is going to be run through a series, I think it's 8 different enzymatic steps, right? 8 different enzymes in here. And we are going to complete the oxidation. We're going to remove the rest of the hydrogens, right? The rest of the electrons. And so what we see here is for each acetyl-CoA, when it goes around the citric acid cycle, we get 2 CO2, right? Because we're going to finish, we're going to completely destroy what's left of this glucose molecule. So 2 carbons came in, 2 carbons have to go out as CO2. We are also going to make, oh, they're doing this in order. Here, see these 2 NADH? And there's one more over here. So 3 NADH, 1 FADH, and 1 ATP. Okay, now the citric acid cycle is much more complicated. Oh, and the important thing to remember here too, right, is that was just 1 acetyl-CoA. And so for a glucose molecule, it has to go around twice. Here is your closer look at the citric acid cycle. So one of the things that I think is most fascinating here is notice this molecule, oxaloacetate. It's a 4-carbon molecule that's willing to bind to acetyl-CoA, right? So each acetyl-CoA, the 2-carbon molecule comes in, and they form a 6-carbon molecule, citrate. Citrate is just another way to say citric acid, which is why this is the citric acid cycle. Okay, and so you do not need to memorize each and every one of these intermediate steps, right? But realize that this is quite complicated. You have lots of enzymes acting here to make this work, right? But again, as we complete the oxidation, NAD and FAD are going to grab those electrons and carry them for us. So again, in the citric acid cycle, per acetyl-CoA, right? For one time around, you get 2 CO2. Ooh. 3 NADH. The nice thing about this picture is they are showing you that those are, coming out at different times. But for us, I think it's fine. Sorry, this is really messy. 3 NADH, 1 FADH. I'm just going to put an F, and 1 ATP. Okay, times 2, because it's going to go around twice. Okay, so you do not need to go further than that. If you go on and take a biochemistry class or something like that, you'll get this. Okay, so we're on to the final step here, oxidative phosphorylation. So oxidative phosphorylation, remember, included the electron transport chain and chemiosmosis. So what we're going to see in the electron transport chain is this is a series of proteins that are embedded in the inner mitochondrial membrane. So the mitochondria actually has 2 different membranes. This inner membrane has the electron transport chain, and we're going to see that the electron transport chain is going to keep, what they're trying to show you here, right, is that we go ahead and we oxidize NADH, right, it goes back to NAD. So it loses its electrons, and it loses them to these proteins, and it just keeps passing them down to lower and lower and lower and lower and lower energy states, right? Which they like, right? You're becoming more stable. Remember this whole free energy thing from the last chapter? We didn't get too into it, but we said, okay, things want to be at that lower energy state. They're more stable. So we pass them along this chain. And at the very end, and this is a key point in aerobic cellular respiration, right, is that it uses oxygen, okay? Oxygen's whole role in this process is to accept the electrons at the end of the electron transport chain. It is the final electron acceptor. It's going to join with some hydrogen protons, and we get water. Okay, and we'll look at that again. Okay, so here is that inner mitochondrial membrane. Okay, and what we're seeing here is the oxidation of NADH, right, becoming NAD. The electrons, I pointed out, are going to get passed through this series of proteins. But notice what happens since we are following hydrogen. Hydrogen's atomic number was 1, which means it has 1 proton. It also has 1 electron. Oddly enough, if you look, hydrogen's atomic mass is just over 1. Most hydrogen doesn't even have a neutron. So when we steal the electron away, all that's left, when you see H+, this is a proton, okay? Inside your mitochondria, you are splitting atoms of hydrogen into individual electrons and protons. Just saying. Okay, so all these hydrogens then are going to be between those membranes, right? This is inside of the mitochondria. This is between the two membranes of the mitochondria. And so what happens is, because oxygen is willing to get reduced at the end of the electron transport chain, as long as there's oxygen, there's like a never-ending kind of place to put electrons. So you just keep reducing and reducing and reducing NADH, FADH, as long as it's getting handed to you. And so these protons start building up in this space between the membranes. And we know things don't like that, right? They want to diffuse, they want to spread out, they want to find equilibrium. But this membrane is impermeable to hydrogen protons, except for at this protein called ATP synthase. Give you 3 guesses what this protein does. Okay, so what's going to happen here is as hydrogen passes back through this membrane, right, it's diffusing, it's going down its concentration gradient, which this kind of annoys me that they call it osmosis and it's not water, right? This is chemiosmosis. It's the diffusion of these hydrogen ions. Anyway, they go through this ATP synthase, and they actually are able to spin the ATP synthase like a turbine. Look at this. So I'll get out of your way. So the hydrogen, as it's coming into the ATP synthase, the way it docks, it actually like starts this rotor going, right? So every hydrogen is essentially fueling the turning of this turbine, right? And the energy coming out of this turbine is then how we put ADP and phosphate together into ATP, because adenosine diphosphate has a negative charge and phosphate has a negative charge. They don't want to go together. We need energy from this turbine to make it happen. So this is weird. Like, so check this out. So you have this whole collection of hydrogen protons up here, right? I'll just put a bunch of pluses for protons, right? You could put H's and pluses if you want, but my writing is not that great. All of these protons are stuck behind this wall. It is like water behind a dam, right? And gravity is pulling it downhill, just like diffusion is pulling these hydrogen ions back into the mitochondria. So now this ATP synthase starts to look an awful lot like a hydroelectric turbine, right? When water is allowed through the dam, it can turn a turbine and we create electricity. What the cell is doing is letting hydrogen turn this turbine and packaging that energy in the form of ATP instead of electricity. And that's pretty phenomenal, okay? So what we end up seeing, right, is, oh, look at that. Now they gave pyruvate oxidation. It's 2-NADH. Anyway, only I would care about this. So glycolysis, right? They're pointing out that you get those 2 ATP, citric acid cycle, you get those 2 ATP, and then all of those reduced molecules, all those hydrogens go to oxidative phosphorylation and are able to supply enough energy to phosphorylate 26 to 28 ATP. now. So then total, they're saying 30 or 32. Don't get frustrated when you see different numbers in different books in different places. So some of this has to do with, so when they're bringing some of these NADH, FADH in, they use a variety of shuttles. You can lose some energy there. It's not, there's like a rounding that goes into this. So it's around 3 ATP that can get phosphorylated per NADH and around 2, but it's like more or less. And this gets really complicated. So don't, biochemistry books will have slightly different numbers. My microbiology book says 36 to 38. Don't let it throw you, but you should realize that almost all of it, these are real numbers, right? 2 ATP, 2 ATP, and then this, there's some rounding error in there. Most of it's coming from oxidative phosphorylation. Okay, so that is aerobic cellular respiration. See, that really wasn't that painful. Okay, in class, we'll try to go through it slightly differently. Watch that video, the BioFlix, hop on Khan Academy, right? Every general biology class in the country, probably the world, is doing this. So there's lots of resources out there. Okay, so then the other thing I did want to mention is fermentation. So if there's oxygen present, right, when we have pyruvate at the end of glycolysis, we're going to send it to the mitochondrion, right, and try to get as much ATP, right, we'll go with 26, 28 with your textbook, out of that glucose as possible, right, because glycolysis only gave us 2 ATP, aerobic cellular respiration will give us the other 28 or 30, right, sorry, 26 or 28. Okay, now, if there's not oxygen present, you can't do it, right? Because if oxygen isn't there to take electrons at the end of the electron transport chain, those electrons back up, we stop being able to oxidize the NADH, and our turbine turns off, okay? So in fermentation, what we're going to see happen is we're essentially just going to go with the 2 ATP that we got out of glycolysis. As a byproduct, so things like yeast are going to produce ethanol, alcohol, right? Our cells will produce lactate, also known as lactic acid, right? Which you feel when you do some sort of anaerobic exercise, right? Where you work out so hard that maybe your cells didn't have enough oxygen to run aerobic respiration, and you had to settle for fermentation. So the thing that happens here that confuses people, so let's use humans, right? So here's lactic acid fermentation. So we are going to do glycolysis, right? We're going to split that sugar. So we start with glucose and we end with pyruvate, 2 pyruvates in the process of glycolysis. And we got 2 ATP and 2 NADH, right? Same as we saw earlier with glycolysis. Now, 2 ATP isn't a lot, but it's okay. The problem is you need to keep splitting sugars, right? And doing it quickly to try to make up for the fact that you're only getting 2 ATP. But what's going to happen is you're going to run out of NAD, right? Because without oxygen, you can't shuttle NADH into the mitochondria and steal the electron from it. And so NADH is just sitting there in that reduced form. So you run out of NAD that you need in order for glycolysis to keep going. So in fermentation, the whole point of fermentation is to regenerate, to remake NAD. So we need to be able to do an oxidation reaction here, right? Steal that electron. Well, look at this. Here's pyruvate, 3 carbons, bunch of oxygens. Here's lactate or lactic acid, 3 carbons, all those oxygens. Oh, and look at this. This didn't have an H, this does. So we are able to oxidize NADH to NAD, right? Regenerating it, rebuilding it for glycolysis to use, as long as we can shuttle that hydrogen somewhere. So we stick the hydrogen on pyruvate, we get lactic acid. Okay, game on, keep going, right? The same exact thing is happening over here, except other organisms, they take pyruvate, right? They do this intermediate step where they get CO2 production, okay, and move to a closely related molecule called acetaldehyde. You don't need to memorize this, right? Basically, they're taking the pyruvate and they are sticking another hydrogen on it to get ethanol so that they can keep doing anaerobic respiration and getting 2 ATP. Because 2 ATP isn't much, but if you can do it fast enough, you'll be all right. That's fermentation. So in lab this week, we'll actually be doing an experiment using fermentation, and we're going to measure fermentation by this CO2 right here, right? How much CO2 is produced in the process. Okay, so then this is what I was mentioning towards the very beginning, is realizing what we just did here is we followed a carbohydrate, right? We took glucose, a sugar, and looked at glycolysis, pyruvate, there we go, pyruvate oxidation, citric acid cycle, yada, yada. Notice you can use amino acids, right? If you rip the nitrogen group off, that's why they're showing you ammonia over here. If you rip the nitrogen group off, it actually looks an awful lot like sugar. And so if it's a 3-carbon, you can feed it in, kind of like pyruvate. If it's a 2-carbon, you can feed it in like acetyl-CoA, and actually some amino acids go directly into the citric acid cycle. Okay, fats. We know that fats are a great way to store energy, right? They actually have over twice as many calories per gram. And so what we do is, remember our triglyceride, right? We had a glycerol and 3 fatty acid tails, right? Glycerol is a 3-carbon molecule, and so we rip that off and feed it in like it's a sugar. The fatty acid tails, remember they were long strings of carbon and hydrogen? We rip them apart 2 carbons at a time, and it looks just like acetyl-CoA, and so we feed it in. And so our bodies can use any of the macromolecules really as a food stock, as a fuel. Now, I would say it kind of looks like this. In like moderate activity, like so you're not doing much. I want to say like, I don't even want to just say moderate activity, like low activity and moderate activity, your body is primarily actually utilizing fats. It's going to save the glucose for the brain. If you're doing intense activity, then you actually need lots of glucose. We don't love to use proteins as a fuel source, but we will if we feel like that is necessary. Okay, one other thing I wanted to point out here, right, is you think about like these are the things in your diet, right? When you eat protein, it gets broken down to amino acids to get digested. And then you can either use it as a fuel or you can run anabolic reactions and make proteins like skeletal muscle or antibodies. Okay. When you eat carbohydrates, you break them down into glucose to absorb them, but you then actually can do an anabolic reaction and build glycogen, a more complex carbohydrate that you're going to store in your liver or skeletal muscle and save for later, right? When you eat fats, you break them down into fatty acids, right? But then you can go ahead and store them as triglycerides if you have too much energy. Heck, if you have too much energy anywhere in here, right? If you can break all these down to acetyl-CoA, and acetyl-CoA, if it's like backing up, there's too much of it, it can't go into the citric acid cycle. What do? You put it back together, 2 carbons at a time, and you store it as fat, right? Because that's a phenomenal storage medium for energy. Okay, so I did want to point out you can use all of these fuels, all of these macromolecules as fuel. And then also just to keep in mind, the food you eat can also be used to run anabolic reactions, as long as you have enough energy. Okay, so then, and here's where, no, that didn't do what I wanted. Again, basically, once you have enough, all they're trying to show here is once you have enough ATP, it's going to tell cells to stop doing glycolysis, right? Hey, why don't you take that glucose and make glycogen instead? And if the citric acid cycle starts backing up, right, there's so much citric acid, that's going to inhibit glycolysis as well. And really, we can take any of this acetyl-CoA and store it as fatty acids. Okay, that is it. I will see you in class.